Animal migration has fascinated biologists and natural historians for thousands of years. Bird migration is highly complex and often involves individuals learning migration routes from family members or conspecifics. For many birds, migration is not learned, but is an innate behavior, and there's strong evidence that part of the behavior has a genetic basis. For example, common cuckoos are birds that are raised by different species, and yet can still migrate to their population-specific winter grounds. Willow warblers from different populations have different migration routes, but hybrid willow warblers migrate across the range of both parents' routes. However, finding a genetic difference between birds with variable migration behavior has proven difficult. The authors of a review paper in Journal of Avian Biology believe that differences in gene expression regulation, rather than differences in gene sequences, lies behind the genetic basis of migration in many species. Stretches of DNA, known as cis-regulatory elements, which lie outside of the coding sections of genes, are often responsible for gene expression. Differences in gene expression have been shown to be associated with whether monarch butterflies are migratory or not. Migratory behavior may also be influenced by epigenetics, which refers to non-coding aspects of genomes, including DNA methylation, chromatin folding, and attachment to the nuclear matrix and the packaging of DNA around nucleosomes. The great diversity of ways in which genetic regulatory mechanisms seem to influence migratory behavior may reflect the need to be able to adapt behaviors more quickly than would be possible by changes to DNA sequencing alone. The authors of this paper believe that different species could have reached similar migratory strategies through different gene regulatory mechanisms via convergent evolution. If epigenetic tools and gene regulation influence the diversity of migration routes and strategies, this may boost the evolutionary potential of migratory birds when faced with environmental challenges.